Perfect. So my name is Naomi de Hart, and I am currently finishing up my master's thesis project in nutrition science at the University of Utah. My mentor is Dr. Drummond, and I just want to say I did my undergraduate degree at Boise State University, where I met Eric Bastian during my senior year, and that is how I got started getting involved with the BUILD program. So I'll jump right into it. So in our lab, we study muscle, and mainly we want to look at how to prevent muscle loss. So in our experience, protein synthesis rather than protein breakdown is the most dynamic variable. It changes throughout the day, and it's largely driven by anabolic stimuli, such as exercise and amino acids coming from foods. Within the amino acids, it's mainly leucine, one of the branched-chain amino acids, that causes this response. It's the chronic daily sums of these negative or positive protein balances that determine whether you maintain, gain, or lose muscle mass, as you can see at the bottom right there. I mentioned that leucine is the main driver of protein synthesis, and this is basically how it works. Let's see if I can. There we go. Perfect. So leucine is right here. So it's the main driver of protein synthesis, and this is basically how it works. Leucine inhibits cestrin, which in turn can activate GATOR2, because usually cestrin inhibits GATOR2, but once it's deactivated, it activates GATOR2. Then this inhibits GATOR1, which then can activate these RAG GTPases. Once this is activated, the GTP will bind to RAG-AB, and the GDP will bind to RAG-CD, and then this all will recruit mTOR to the lysosome right here. Since mTOR controls the anabolic and catabolic signaling of skeletal muscle, the ingestion of amino acids in turn leads to the regulation of muscle hypertrophy and muscle loss. mTOR increases protein synthesis by enhancing mRNA translation through the phosphorylation of its downstream targets, P70S6K1 and 4AB. Since I just mentioned that it binds to the lysosome, or mTOR binds to the lysosome when it's activated, it seems like the lysosome is the best location for mTOR to be in order to increase protein synthesis. So I think it's important to understand what this translocation actually means when looking at protein synthesis directly. In this graph, you can clearly see a positive correlation between protein synthesis on the y-axis and mTOR and LAMP2. And LAMP2 is a stain for the lysosome. So this is the co-localization of mTOR to the lysosome, so when it binds to the lysosome. So there's a positive correlation between those two things. Thus, identifying where mTOR is within the cell is a good readout of muscle anabolic processes following protein-dense food ingestion. Similar responses to what I just showed has been shown after the ingestion of other whole foods or protein-dense foods. This study in particular looked at 36 grams of protein coming from pork, and this accounted for about 3 grams of leucine. And as you can see here, I can actually not see it, but the branched-chain amino acid levels in the serum were elevated for almost the entire 5 hours post-ingestion. And then also here is a graph that shows protein synthesis on the y-axis, and it's also increased after the ingestion of this protein-dense food. So for my project, we wanted to look at cheese, in particular cheddar cheese. This whole food is more likely to be consumed by the general population, by the public, than other protein fractions. About 11 pounds per year per capita of cheddar cheese is consumed in this country. It's high in leucine content. About 10% of its total protein content is leucine. But surprisingly, there is not a whole lot of information on the blood amino acid kinetics, nor on the muscle anabolic response following a single dose of cheddar cheese in humans. This brings us to the purpose of the study. So for the first aim, we wanted to determine the blood branch chain amino acid and muscle mTOR signaling response to the ingestion of cheddar cheese, accounting for 20 grams of protein. And then for the second aim, we wanted to compare the blood branch chain amino acid response and mTOR signaling response to the ingestion of an isonitrogenous and semi-isocaloric amount of milk. And then here at the bottom, you can see the nutrient content of the two products that we used in the study. We matched the products for protein. 20 grams of protein was chosen because this has been shown to maximally activate protein synthesis in humans. And milk was chosen as a comparator since it's also very high in leucine content, also about 10% of its total protein content is leucine, and its amino acid kinetics are very well characterized. And what I didn't show here, but I do want to mention, is both products had about 2 grams of leucine. Then the experimental design of the study. So here's the time course of the study. After recruiting 12 males and 12 females and screening them, enrolling them, they came to us for the study. There's 2 study days, study 1 and then study 2. They recorded their food prior to both study day 1 and study day 2 for us to get an idea to what they eat on a regular basis and how much protein they consume. Then we also did a body composition test to characterize the population that we're studying and to see how much lean mass they have and their percent body fat. And then step activity is also mentioned here. We also asked participants to keep track of their step activity throughout the study, so in between study days 1 and 2. And then during the study days, this is what happened. The participants came into the research facility at 7 a.m. after an overnight fast. After getting settled in, we took a baseline blood draw and a baseline muscle biopsy, which is right here, from the thigh muscle. And then we gave them the product, cheese or milk. It was randomized, so first they did it one product, and then the second study they did it the other products. And then we took blood draws for the consecutive 5 hours after that. And then we took muscle biopsies 1 hour and 3 hours post-ingestion. So this whole process this day was the exact same for the second day, but with the other products. And then we also gave them a research meal prior to both study days to standardize the meal the night before. Here's a table with the subject characteristics. So as I mentioned, we recruited 24 people. So 12 of them were males and 12 of them were females. Healthy young adults, 27 years of age on average, BMI at 23. And then their normal dietary protein intake was above the RDA, and they were normally active. Then moving into the results. First, I wanted to show the essential amino acids and the non-essential amino acids in the serum post-ingestion. As you can see here, milk spikes right after the ingestion and slowly moves its way down almost towards baseline over the time course of the study, which was 5 hours. While cheese shows a prolonged increase in amino acid levels in the serum. And then on the right side, you can see the area under the curve, which shows that there is not a difference between the two products when you look at amino acid levels in the serum. And same goes for the non-essential amino acids. And as I mentioned, the main drivers for protein synthesis are the branch chain amino acids. So here they are. And just like the essential and the non-essential amino acids, milk shows up a little earlier than cheese, but comes down quicker. And also here, total area under the curve for the entire time period of the study does not show a difference between the products. And then when you break it down in the individual branch chain amino acids, there's the same trend going on here, and there's no significant difference between the products. As far as muscle analyses, as I mentioned, we took muscle biopsies, and then one of the analysis is immunohistochemistry. So after we take a muscle biopsy, we separate the tissue into different pieces for different analyses. And one of them is this analysis. For this analysis, we always try to get a nice and clean looking piece. Why? Because we want to section it and want to make it look good on the piece of glass that we put it on. So we section it, and we do this in, it's basically a tiny meat slicer that is cooled to -20 degrees Celsius. After doing so, we put the little slices on a piece of glass and let it dry. Then the staining process starts, and we use different fluorescent antibodies to stain the border, the cell membrane, the nuclei, and the proteins of interest. In this case, mTOR and LAMP2. This way you can see the local effect in muscle from food ingestion. After we do that, we image the slides and get very pretty images. At least I think so. And then we analyze the data at the end. So here are some of the images that I took. On top left, these are nuclei. This is the cell membrane. And then this is mTOR. So these are the fluorescent antibodies that bound to the mTOR in the muscle. And then here's LAMP2, so all the lysosomes. And then when you put it all together, this is what you get. And what we actually want to look at is where it co-localizes. So wherever mTOR and LAMP2 overlap. With the white arrows, you can see that over here. We're in the process of analyzing the data right now. And then another analysis that we do with the muscle is Western blots. This gives us the opportunity to look directly at how much of certain proteins downstream from mTOR are activated. So I mentioned before that downstream of mTOR is P70S6K1, and we did Western blots for that. And as you can see here, after the ingestion of both cheese and milk, there's more P70S6K1 phosphorylated, which means that mTOR activated that. And then when you look at downstream from P70S6K1, there's ribosomal protein S6, and also that is more activated post-ingestion, as you can see right there. And then the final steps for my project are to look at the insulin levels and the free fatty acid levels in the serum. And for the muscle, finalizing or finish analyzing the data from both the immunohistochemistry and the Western blots. And then we will also compare the group of males and the group of females to see if there was any sex differences. And with that, I want to thank Bill Derry, Glamia Nutritionals, my committee members, and the Drummond Lab. Are there any questions?
Any questions? Remember to unmute your mic.
So this is Don McMahon. So nice presentation, Naomi.
Thank you.
So I have a kind of sense of general question related to overall dietary habits. In the sense that show the graph where you see this increase and decrease based upon breakfast, lunch, and dinner. You don't have to go back to it. But my question is that
This one, right?
Yeah, no, this is just in general where you show switching from the anabolic to catabolic state
Yes.
when you have a meal. And so you think that individuals would be better off if they had, if we were part hobbit, for example, and we had breakfast, second breakfast, 11 C's lunch, and whatever. It had more meals during the day and smaller meals rather than large meals. My question.
Okay, so the question is, if small meals throughout the day are better than large meals, right?
Yeah.
So if you overstimulate this pathway, then you become kind of resistant to it. So in my opinion, yes, I do believe that small meals throughout the day will help you maintain a positive muscle protein synthesis balance, if that makes sense.
And you, as a follow-on from that, you mentioned in your talk about why you chose 20 grams of protein. I think that's because that's where you see an effect at. So is there a minimum amount of protein you need to have in a meal in order to see this going into the protein synthesis state rather than breaking down?
That's a good question. So the 20 grams is to maximally activate protein synthesis. You see an activation if you just give straight up leucine, you or part of a protein matrix, then you see an activation with like way less than 20 grams. But since we wanted to maximally activate it, that's why we use 20 grams.
So does that mean for normal human beings, not those ones who are trying to purposely add bulk muscle, that more than 20 grams of protein is wasted?
Yes, it has been shown that more than 20 grams per serving does not increase protein synthesis more than just 20 grams per serving, per meal, basically, yes.
So in light of what you found with the milk versus cheese, I imagine that 20 grams comes from an acute ingestion of very available amino acids or proteins. So if the cheese form of the the food that you're ingesting, it has an effect on how long it takes for that amino acid levels to increase and then kind of maintain and decrease. You think that you would see, compared to a milk where 20 grams of protein, or whey protein where 20 grams of protein may be the maximum your body can cope with at one time, if you were taking say 30 or 40 grams of protein in cheese, which is a lot of cheese, I understand that. 40 grams would be a quarter of a pound. But some of us have been known to eat that on a pizza, maybe at the same time. Do you think that maximal level would increase if you had a slower release of the amino acids?
What's the question? Sorry.
Yeah, so you said 20 was the maximal, which I assume, meaning once you take more than 20, you don't get any increase in fat. But if you consume it in the form of cheese, and seeing that's a much slower release, do you think that you could benefit, if you had 30 grams, you would actually get a positive benefit from that extra 10 grams?
That is a very good point. Maybe, yes. That is a very good point. Since it's released slower and you don't see that spike, then yes, that might be what could happen, yes.
Questions? Okay, thank you, Naomi.
Perfect. Thanks.